we now talk about the two different isomeric form of glucose. We can have alpha glucose. You can also have beta glucose. So the alpha glucose and beta glucose depend on the orientation of OH groups with the anomeric carbon. But in this case, we also have D-glucose and alpha glucose. So what is the difference between D-glucose and alpha glucose is actually based on the position. It's based on the position of the OH group. So in carbon 5, the OH group is by the what? Is by the right. While for the L-glucose, the OH group is by the left. And that is what differentiate D-glucose and alpha glucose. And D-glucose is the most abundant, or is actually the class of the glucose that are usually found in the human system. So we don't have L-glucose, unless if we actually take them or if we get them from foreign organisms or maybe from other foreign substances. You cannot even digest alpha glucose because you don't have the enzyme that can do such. But D-glucose is the class of the carbohydrate that are actually found in the living system, especially human being. We have D-glucose, we have abundant D-glucose. And L-glucose, we don't have it. In fact, even if we have it, our body cannot utilize it because we don't have enzymes that can take care of that. So now back to the issue of the chiral carbon. Look at it. This is the carbon. And if you look at it, you see that we have X, Y, W, Z. You see they are all different. So that is why they are called chiral center. The center is the center where a carbon attached to 4 different groups. And such kind of carbons is called chiral center, or that center is called chiral center. So the presence of asymmetric carbon atoms in a compound give rise to the formation of isomers of that compounds. So you see, the presence of the asymmetric carbon atoms in the compound, it gives rise to the formation of isomers of that compound. So because we have this, that is why even in the case of the isomers, we have L isomer and the D isomer. So this presence of asymmetric or chiral carbon leads to the formation of isomers in a compound. So now stereoisomerism. What is stereoisomerism? Stereoisomerism are compounds having the same structural formula, but different in spatial configuration. Different in spatial configuration are known as stereoisomerism. So the spatial configuration, it means just a spatial orientation. The compounds, they are the same. Their structural formula are also the same, but only differ in the orientation of a particular group. That is stereoisomerism. And the number of possible stereoisomers depend on the number of asymmetric carbon atoms. So we use this formula 2 power n. I talked about this on a previous slides. And the formula is called Van Hoff. Okay, the formula is actually used according to Van Hoff's rule, where n indicates the number of asymmetric carbon atoms. So now we are going to look at D and L isomers. So the orientation of the hydrogen and OH groups around a carbon just adjacent to terminal primary alcohol group, e.g., carbon atom 5 in glucose, determine the family to which the sugar belongs. Remember also in this case, I told you it's actually based on the orientation of hydrogen and OH group. So if you actually check from our previous slides, you will be able to observe that the orientation of OH group at carbon 5 from the right give or lead to what you call the D isomer, and then for the orientation of OH group by the left leads to the formation of what you call the D isomer. So when the OH group on the carbon 5 is on the right, look at it, when the OH group on this carbon is on the right, the sugar is a member of D series. And when it is on the left, it is a member of the L series. So the distribution of the OH and hydrogen groups in the other carbon atoms in a molecule is of no importance. So you see, the orientation of the OH group and the hydrogen atoms in the compound are always considered at carbon 5 of glucose. So, but for the orientation of other sugars, of other carbon atoms, it's actually not depend. It's not actually depend on the other carbon atoms. So they are of no importance. They are of no importance. So now we are going to look at the different compound and how this compounds from different D and L series. So if OH group on the bottom, chiral center points to the right, the sugar is D-glucose. So let's look at this. This is the OH group. This is the OH group, and it is on the bottom of the chiral carbon because this is the chiral carbon. So it is at the bottom. It is at the bottom of the chiral carbon. So as a result of that, we have D sugar. And look at also this one. We also have D sugar in the case of D-glucose. But look at this. This one is also a chiral carbon. So we have OH group by the left. So it is now considered as L series. Then for the L ribose, look at the OH group. The OH group is also by the left. And this one is by the right, and that is why it is D-glucose. So the majority of monosaccharide occurring in the body are the D series. Remember, I said this. Most of the sugars that are found in human or in the body of human being, they are in the form of D series. Okay? So this is how they look like. Enantiomers, they have mirror image. They have mirror image. So look at, this is the L-glucose and this is the D-glucose. So L-glucose is a mirror image of D-glucose. That is the meaning of the mirror image. They are enantiomers. So those compounds that have mirror image with one another are considered as enantiomers. So now the next thing that we are going to look at is optical activity or optical isomerism. So the presence of symmetric carbon atoms also confers optical activity on the compound. When a beam of polarized light is passed through a solution exhibiting optical activity, it will be rotated to the right or left in accordance with the type of compound. That is the meaning of optical isomer. So those compounds that actually rotate, those compounds that can rotate to the right or left based on the polarized lights are considered as the optical isomer. So those that can rotate by the right side are called dextrorotatory, and those that can rotate to the left side are called laborotatory. So now let's look at the compound here, L-alanine and D-alanine. So what is the difference between L-alanine and D-alanine? Look at it in the case of L-alanine, look at the CH3 here. The CH3 in the case of L-alanine is at the right. So it is actually at the left sides, and that for the D, it is at the right sides. It is at the right sides. So those, you consider the CH3 as a reference, as a reference compound. So if you look at it, you can see that in the case of dextrorotatory, it means that are those that rotate by the right, and those that rotate by the left is called laborotatory. And That is the D-alanine, and that is for the L-alanine. And in terms of the isomer, the L-alanine have S isomer, S enantiomer, and then the laboratory, which is the D, is having the R isomer. So I think in this point, the most important thing that we should all know is just about the orientation. It's just about the orientation of the carbon, of a particular reference compound. Like for example, in this case, I took CH3 as a reference compound. And if you look at it in the case of L-alanine, the compound is actually rotates to the right sides, while in the case of the D-alanine, the compound rotates by the left sides. Okay. So now we are now going to look at racemic or DL mixture. So when equal amount of dextrorotatory and laboratory isomers are present, then the resulting mixture has no optical activity. And those kinds of mixture are called racemic mixture. So when you have equal concentration of dextrorotatory and laboratory isomers, so the resulting compound do not have optical activity. They don't have optical activity, and they are also known as racemic mixture. It means that when you have 50% of the R lactic acids and 50% of the S lactic acid, or 50% of D and L lactic acid, it means that there is no optical isomerism. And those resulting mixture of equal amount is considered or is called racemic mixture. Okay. Then the next thing is Epimers. Epimers is when you have two sugars which differ from each other only in the configuration around a single carbon atoms. They are called Epimers. They are called Epimers. It means that all those sugars that have, so that is Epimers. So it means that when you have two sugars that are differ from each other only in configuration around a single carbon atoms are called Epimers. So now let's just look at these two different compounds. Look at this. This is a compound. Look at D-mannose and D-galactose. What is the difference between D-mannose and D-galactose? D-galactose, D-glucose, and D-mannose are Epimers of each other. So for the D-mannose and D-glucose, there are Epimers. There are carbon 2 Epimers. Look at it. At carbon 2 here, the OH group here is at the left. Sorry, is at the right. At the right in the case of OH group, in the case of mannose, the OH group is by the what?
Is by the right.
Is by the right. What in the case of
D-glucose is by the left.
So that is to tell you that at carbon 2, it's just only this OH group. If you look at it, all other groups that attach to the mannose and the glucose are the same. The D-glucose and D-mannose are all the same. The only difference is just the orientation or the configuration of OH group around carbon 2. That's what makes them differ. And in the case of D-glucose and D-mannose is also the orientation of OH group. And apart from that, the D-glucose and D-galactose, they are all the same. So it's just only the difference of the orientation of OH group at carbon 4. And that is what makes them differ. So ladies and gentlemen, this is what you call the concept of epimers. Are the compound that have the same molecular structure, the same formula, the same number of carbon atoms, the same number of hydrogen, everything are the same, but only the difference in the orientation is just the difference in the orientation of carbon atoms around one particular carbon atoms. And that what make them differ from one another. That is epimers. And then the next thing is enantiomers. That is anomers. We are now going to look at the anomers. We have what you call anomeric carbon. So which one is anomeric carbon here? The anomeric carbon is carbon 1. So carbon 1, in this case, form a ring structure with carbon 5. It forms a ring structure with carbon 5, and it's called anomeric carbon. And of course, remember, we also talk about the alpha and alpha and beta glucose. So the alpha and beta glucose is just the difference in terms of the orientation of OH group. That's what make alpha glucose and beta glucose differ from one another. So in the case of the alpha D-glucopyranose is just that the OH group is pointing down for alpha. Is pointing down to the anomeric carbon. Then in the case of beta D-glucose, the OH group is pointing up. It's pointing up to the anomeric carbon. So you should all know this. Okay. Then the next thing is mutar rotation. Mutar rotation. Okay, ladies and gentlemen, I think let me just stop here for this. Then we are going to rejoin. But before we rejoin, you can ask questions if you have any. So the next thing that we are now we are going to look at disaccharides. Disaccharides. So disaccharide is when you have two monosaccharides that are combined together by a glycosidic or by glycoside linkage. They are considered as a disaccharides. So when you have those two different monosaccharides, joined together by what we call glycosidic bonds, they form what you call disaccharides. And we have disaccharide of biological importance. This disaccharide of biological importance, we have sucrose, we have maltose, and then we also have lactose. So we are starting with sucrose. A sucrose, it is the sweetest agent, sweetening agent, known as cane sugar. So that is to tell you that the sugar that are found in sugarcane, is sucrose. And it is present in sugarcane and various fruits. Sucrose contains glucose and fructose. So it means that the sucrose is actually a combination of glucose and fructose. So remember, glucose is a monosaccharide, fructose is a monosaccharide. So when you have these two different monosaccharides joined together through glycosidic linkage, they form the sucrose. And sucrose is a non-reducing sugar, and it will not form oxazole. This is because the linkage involves first carbon of glucose and the second carbon of fructose. And the free reducing groups are not available. So when we say a sugar is a non-reducing sugar, it means that it's a sugar that do not have free aldehyde or ketone functional group, because both the carbonyl carbon are involved in the formation of the glycosidic bond. So that is glycosidic, that is the sugar that actually considered as a non-reducing sugar. So that is why we said in the case of sucrose, remember the first carbon of glucose is the anomeric carbon, or is actually the carbonyl carbon, while the second sugar of fructose is also the carbonyl carbon, the keto group. So they are all involved in the formation of the glycosidic bond. And that is why there is no any free reducing groups left. So as a result of that, they are considered as a non-reducing sugar. So ladies and gentlemen, sugars can also be classified as reducing sugar or non-reducing sugar. So any sugar that have free reducing group, they are considered as, if those sugars that doesn't have free reducing groups, they are considered as non-reducing sugar. While all sugars that have the free reducing groups, are considered as a reducing sugar. An example of this non-reducing sugar, the example of the reducing sugars, all monosaccharides are reducing sugar because they have free aldehyde or ketone functional group. But in the case of sucrose, it is actually a non-reducing sugar because none of the reducing groups is available from both the glucose and the fructose. So this is the example. This is how it looks like. This is alpha.
So what is that ozone?
You said?
Ozone, you have already mentioned about ozone. What is that ozone?
Yes, what we call an ozone is when you react the reducing sugar, react this non-reducing sugar with some chemical. So they do not form an intermediate called ozone. So, but because the reducing sugar doesn't have, because sucrose is a non-reducing sugar, it doesn't have the free aldehyde or ketone functional group. So that is why it doesn't react. So therefore, the ozone will not be produced. So this is how we have the examples of the sucrose. This is the carbon 1 of glucose, and this is the one here, and this is the C2, that is carbon 2 of the fructose. So they attach by glycosidic bond. So at the D-glucose, look at it, this is the anomeric carbon. This is the anomeric carbon. So it is alpha D glucose that actually form a bond with the beta D fructose. So that is why we have alpha D glucosyl beta D fructosides. And this is the same thing as sucrose, is the same as sucrose. So the next thing that we are now going to look at is lactose. Lactose is also a sugar that is present in the milk. Like in the breast milk, this is the sugar that usually found. And it's also called milk sugar. And it is a reducing disaccharide. It is a reducing disaccharide. Because on hydrolysis, lactose yield glucose and galactose, and beta glycosidic linkage is present in lactose. So the anomeric carbon atoms of beta galactose is attached to the 4th hydroxyl group of glucose through beta-1,4 glycosidic linkage. So that is to tell you that the anomeric carbon of the glucose is still available. It's not involved in the formation of the glycosidic bond. And that is why lactose is also considered as a reducing sugar, because there is a free aldehyde group from the glucose. And that is why lactose is considered as a reducing sugar, and it's made up of glucose and galactose. So this is how it is. We have galactosyl beta-1,4 glycosidic bond. So you see here, the anomeric carbon is still available here. It's still available, the carbonyl carbon. It means that there is a free aldehyde, or there is a free carbonyl group here. So that is why it is considered, or there is a free reducing group. That is why it's considered as a reducing sugar. The maltose. Maltose is also a reducing disaccharides, and it contains glucose residues. It means it's 2 glucose that form maltose, and they form alpha-1,4 linkage, that is the anomeric first carbon atom of one glucose is combined with a fourth hydroxyl group of another glucose through alpha glycosidic linkage. So that is what we have in the case of maltose. So maltose and lactose, they are all examples of reducing sugar. And why are they reducing sugars? Is because they have free aldehydes. They have free aldehyde or keto group. So ladies and gentlemen, I'm now going to stop here and we are going to rejoin again, unless if you have questions, because we are going to start discussing on polysaccharides.